Hi everyone, welcome to today's webinar on behalf of Biocytogen. My name is Xiaoxuan and I'm the Marketing Executive at Biocytogen Beijing site. I'll be your moderator for today's session. We are thrilled to have you join us as we explore humanized FcRn mice as a preclinical tool for antibody PK/PD and therapeutic development. Today, we are pleased to introduce one of our experts who will be sharing insights that can support and enhance your ongoing research programs. Now, it's my pleasure to introduce today's speaker, Dr. Meiqi Zhang. Dr. Zhang earned her Ph.D. at Peking University, where she focused on the mechanism of copper chaperone proteins and the use of copper chaperone antagonists in tumor metal immunology. She then joined Biocytogen as a product manager in the BioMice product department. Dr. Zhang leads the development of humanized mouse models for neurological disorders, as well as models that support large molecule drug penetration across the blood-brain barrier. Her work has enabled a wide range of humanized mouse models to be applied across multiple therapeutics areas, including Alzheimer's disease, Parkinson's disease, ALS, epilepsy, and other rare diseases. So now I would like to invite Dr. Meiqi to begin her presentation. Participants may ask their questions in the Q&A box or drop us an email, and our representative will address the questions by email respectively. Thank you, and I hope you enjoy the presentation. Let's welcome Dr. Meiqi.
Hello everyone, thank you for joining today's webinar. I'm Meiqi, I'm a product manager of Biocytogen and majoring in development of humanized mice models. Today I will talk about how humanized FcRn mice can serve as an effective preclinical tool for antibody PK and PK study, and these models used in the therapeutic development. And here is today's contents. First, I will introduce FcRn function and the need for humanized mice models. And second, I will introduce the development and validation of humanized FcRn mouse models in Biocytogen, including the humanized FcRn mice in the B6 background, in the rats, and in an immunodeficient background. And I will also introduce a double humanized mice, which is the humanized ALB and humanized FcRn mice. And the last one is the summary part. And first, I will introduce FcRn function and the need for humanized models. FcRn is mainly found in endothelial and epicellular cells. Its key job is to protect IgG antibodies and albumin from being degraded, which helps prolong their half-life in our body. It works in a pH-dependent way, and acidic pH, like inside cells. FcRn binds to IgG, facilitating its transport and recycling. But when the pH returns to neutral, like in blood, it releases the antibody from FcRn into the extracellular fluid. And this recycling system is what keeps antibody levels stable over time. So why do we need humanized FcRn? First, FcRn function is species-specific. Human IgG binds to mouse FcRn through different amino acid residues, leading to PK data that doesn't align with human results. And second, FcRn humanization is necessary for the evaluation of antibodies with Fc modifications, such as YTE mutations. This mutation binds more strongly to mouse FcRn at neural pH, like pH 7.5 compared to the wild-type IgG, but not to human FcRn. And this increased affinity hinders efficient serum release, offsetting the benefits of enhanced binding at acidic pH, like pH 6, resulting in reduced serum concentrations in the mice. So this is why FcRn humanization is essential for translational PK and PK studies. And next, I will introduce some humanized FcRn mouse models developed and validated by Biocytogen, including humanized FcRn mice in the B6 background, in the rats, in an immunodeficient background, and also in double humanized mice. And first is the humanized FcRn mice on the B6 background. The targeting strategy of this mice is the human full-length FcRn cDNA was inserted into the mouse FcRn exon 2 of the wild-type mice, and we replace the mouse FcRn with human FcRn. For this mice, we also have some model validation data. From the mRNA expression data, we can see that the mouse FcRn mRNA was knocked out in the homozygous mice, and only the human FcRn mRNA can be detected in the homozygous mice. And this sequence was confirmed by Sanger sequencing. And for the protein expression data, we have detected FcRn expression in various tissues, including the heart, liver, spleen, lung, kidney, muscle, stomach, colon, testes, and eyes. And we can see that the mouse FcRn can only be detected in the wild-type mice, and the human FcRn can only be detected in the homozygous mice. We also detect various mouse immunoglobulins in these mice, including mouse IgG, IgM, and IgA. We also detect subtypes of mouse IgG, including IgG1, IgG2b, IgG2c, and IgG3. From the data, we can see that the mouse IgG levels in the homozygous mice was lower than that in the wild-type mice, because mouse IgG bind less efficiently to the human FcRn, and they are cleared faster, while IgM and IgA levels in the homozygous mice was as the same as that in the wild-type mice. Next, we evaluate PK data of antibodies with Fc modifications. For example, the YTE mutation, which can enhance FcRn binding and prolong antibody half-life. In the humanized FcRn mice, we can see that YTE-modified antibodies circulate longer than wild-type antibodies. But in contrast, this effect doesn't appear in the wild-type mice. So only FcRn humanized mice can accurately evaluate the PK characteristics of YTE-mutated antibodies. And this data also showed that our humanized model correctly mirrors human antibody and FcRn interactions. Here is also some other application cases provided by our clients using FcRn humanized mice in evaluating YTE-mutated antibodies PK value. And we can also see the same results that YTE mutation enhances FcRn binding and prolongs antibody half-life. Using these humanized mice, we can also compare antibody half-lives across species. We collect different antibodies PK data in human and monkey, and we also perform PK study using humanized FcRn mice and wild-type mice. And the data showed that the correlation between human and humanized FcRn mice is similar to monkey and better than the wild-type mice. And humanized FcRn mice can mimic antibody processing in human and monkey and provides reliable PK prediction at a much lower cost and with higher throughput than the non-human primate studies. In PK study, target-mediated drug disposition or TMDD effect will also affect drugs' PK patterns. Some antibodies bind very tightly to their targets, and this can alter their distribution or clearance. The FcRn pathway only explains part of the PK behavior, and TMDD adds another layer of complexity. According to this, we have also crossed the FcRn humanized mice with other target humanized mice, such as CD40, CD98 heavy chain, and TF1 humanized mice to better evaluate PK of anti-human target antibodies. For example, in CD40 and FcRn double humanized mice, we assessed the PK values of two anti-CD40 antibodies produced in-house. We can see the half-life of these two antibodies in the double humanized mice is shorter than that in the wild-type mice and the single humanized FcRn mice. And this is because the antibodies bind to targets, leading to a higher rate of internalization and degradation of the antibody, resulting in accelerated clearance and a shorter half-life of the antibody compared to the situations without TMDD effect. So the double humanized mice can better reflect in vivo antibody concentration changes and suitable for evaluating the PK of anti-target antibodies. The humanized FcRn mice model can also be used to evaluate FcRn blocking drugs, therapeutic agents that intentionally disrupt FcRn and IgG interaction to lower pathological IgG levels. This approach is used in the treatment of autoimmune disease and certain antibody-mediated disorders. In such disease, there is an abnormal elevation of pathological IgG antibodies that can cause tissue damage and disease symptoms. By blocking the interaction between FcRn and IgG, the aim is to reduce the elevated levels of these pathological IgG antibodies and reducing the harmful immune response mediated by these antibodies, then relieve symptoms. So we also have applied this humanized mice in this section. The single dose of FcRn blockade reagents led to decrease the IgG concentrations, and these mice can be used in drug efficiency assay in inhibiting human FcRn and IgG interaction in some autoimmune disease. Here is the summary of these mice. The targeting strategy of these mice is target knock-in of human FcRn CDA into a mouse FcRn genomic with endogenous promoter and mouse B2M. And we have validated mRNA and protein expression in various tissues. We also test the FcRn function in IgG salvage and trafficking. It mimics human IgG half-life and clear differential of Fc variants antibodies. In the PK study, we have tested monoclonal antibodies with Fc modifications and bispecific antibodies. And these mice correlates with human PK values with low variability. And these humanized FcRn mice have been granted patents and is widely used in academic and industry. And here are some publications using our humanized FcRn mice. And next, I want to introduce humanized FcRn rats. The advantages of rats over mice are rats have a bigger body size and a higher blood volume than mice. This allows researchers to collect larger volumes of biological samples, such as plasma, serum, and CSF in a single draw. It's particularly beneficial for PK study that require multiple analytical from the same time point sample without needing to pull samples from multiple animals, which can introduce variability. For the humanized FcRn rats, the gene targeting strategy is as the same as in the mice, which is the human full-length FcRn CDA was inserted into the mouse FcRn exon 2 of the wild-type rats. We replaced the rat's FcRn with a human FcRn. And this rats, we also have some model validation data. This is the protein expression data. We can see that the mouse FcRn protein was only detected in the wild-type rats, and the human FcRn protein was detected in the homozygous FcRn humanized rats. And using this FcRn humanized rats, we can also perform PK study. For example, in this experiment, we assess an anti-VEGF-A antibody bevacizumab. Rats allow for continuous collection of blood samples for ELISA testing. Results showed that the lifetime of this antibody in humanized rats was shorter than that in the wild-type rat, which is consistent with data in mice. We think humanized FcRn rats can provide PK profiles closely aligned with human data, confirming their translational value. And next, I will introduce humanized FcRn mice on an immunodeficient background. One major problem in PK studies is the formation of anti-drug antibodies, or ADA, which is when exogenous therapeutic proteins enter the body. The the immune system recognizes them as a non-self and produce anti-drug antibodies, then accelerate drug clearance or reduce its efficiency. This is the mechanism of antibodies production. When the antibody is administered, it undergoes endocytosis by antigen-presenting cells. Then the cells will present the antibody-derived antigen to the TCR naive CD4 positive T cells. Then, along with cytokine signaling, the naive T cells was activated and in turn interact with the naive B cells and secrete some cytokines. These cytokines then stimulate B cells activation and differentiate into the plasma cells, then produce and release anti-drug antibodies. So to eliminate ADA effect, CD4 positive T cells that present the antibody-derived antigen or B cells that finally produce the anti-drug antibody should be blocked. So we humanized FcRn on the immunodeficient background mice, which may lack the T cells and B cells. One of these is on the NTG mice, which is also developed by Biocytogen, lacks functional T cells, B cells, and NK cells. And we also have other immunodeficiency models, such as REG2 knockout mice and CB17 scaled background mice. These models allow clean ADA-free PK profiling of human antibodies and their Fc engineered variants. The targeting strategy of humanized FcRn mice on the NTG background is as the same as that in the mice or in the rats, and we also have some validation data about these mice. For example, the protein expression data showed that mouse FcRn protein was only detected in the wild-type mice, and human FcRn protein was only detected in the homozygous humanized mice. Using this NTG background humanized FcRn mice, we can perform PK study without considering ADA effect. In these mice, the half-life of antibody with YTE or LS mutations are longer than that of the wild-type antibody, and this modification of YTE or LS mutation can prolong antibody's half-life. This result is as the same as in the humanized FcRn mice on B6 background. However, in the wild-type mice, the conclusion is exactly the opposite, which proves the inaccuracy of evaluating PK using wild-type mice and the necessity of using FcRn humanized mice. However, it should be noted that when conducting PK studies, mice on the NTG background are not the optimal choice either. Although such mice can indeed eliminate the effect of ADA, but literature reports that the NOD scaled mice have a faster antibody clearance rate when used it in the PK studies. And the NOD scaled mice is similar to our NTG mice, while pure scaled mice are a better choice. So we also have corresponding humanized mice available for selection. And the last model is a double humanized model, which is the humanized ALB and the humanized FcRn mice. Albumin, like IgG, is protected by FcRn and plays an important role in extending the half-life of smaller therapeutic proteins, such as nanobodies or peptide conjugates. Albumin can bind to FcRn in the acidic environment of endosomes, and this binding protects albumin from being degraded in the lysosomes. Instead, albumin is recycled back to the bloodstream by FcRn, thereby prolonging its residence time in the body and increasing its half-life. So we also developed the ALB and FcRn double humanized mice by secondary gene targeting of ALB on the humanized FcRn mice. The ALB humanization strategy is the coding sequence of the full-length human ALB protein was inserted to replace part of the mouse ALB exon 1. In this mice, we also have some model validation data. For the ALB detection, we use the ELISA method, and we can see that the mouse ALB can only be detected in the wild-type mice, and the human ALB can only be detected in the homozygous humanized mice. And for the FcRn detection, we use the Western method, and we can see that the mouse FcRn protein can only be detected in the wild-type mice, and the human FcRn protein can be detected in the homozygous humanized mice. Using this double humanized mice, we also have PK study data. We test an anti-TNFA nanobody. The T-half in the humanized mice was longer than that in the wild-type mice, demonstrating albumin's protective effect. And this model is an ideal model for developing albumin binding or albumin fusion drugs. The last one is the summary part. To summarize, humanized FcRn models provide a range of powerful tools for antibody and protein drug development. We have humanized FcRn mice on the B6 background and BBB background, and we also have humanized FcRn rats. These mice or rats can better assess the PK of human therapeutics and optimize antibody half-life via Fc region modifications, and as well as assess the efficiency of IgG blocking therapeutics. Besides the humanized mice models on the normal background, we have also humanized the FcRn on some immunodeficient background, such as the NDG humanized FcRn mice. And these mice can use in the minimize ADA impact for PK study. And we also have FcRn and albumin double humanized mice. These mice can better reflect PK of albumin-based nanobodies or fusion proteins. Besides this, we have other multi-target humanized mice by crossing the human FcRn mice models with other target humanized models, such as humanized CD40, CD19 heavy chain, and humanized TF1. And these mice models can use to mimic target antibody PK and evaluate the TMDD effect. Together, I think these models can help bridge the gap between animal data and human outcomes. And further explorations may be needed. For example, one exciting new area is FcRn-based sweeping antibodies. These are engineered antibodies that capture and clear soluble antigens from circulation through the FcRn's recycling pathway, and they are being actively explored for autoimmune and inflammatory diseases such as the asthma, IBD, RA, and SLE. And FcRn humanized models are essential for testing their PK and mechanisms. FcRn humanized mice are also valuable for studying brain shuttle antibodies, such as those targeting TFR1, IGF-1R, or CD98 heavy chain. These antibodies use receptor-mediated transcytosis to cross the blood-brain barrier, and we have a series of animal products. Using humanized FcRn mice allows researchers to measure realistic PK profiles and optimize dosing strategies for central nervous system delivery. All these models come from Biocytogen's BioMice platform, which now includes more than 4,000 gene-edited mouse models for new drug development. And these models are used across preclinical research and clinical research, from antibody discovery and PK/PD studies to reproductive toxicity testing and combination therapy evaluation. And this is all the contents. Thanks for listening.
Thank you for your time and thank you for joining this webinar by Biocytogen. That's all for the presentation by Dr. Meiqi. If you have any questions to us, please drop us an email at info@bbctg.com.cn and our representative will reply to you shortly. You may also scan this QR code to know more about Biocytogen from our official website. Thank you once again and I hope to see you next time. Goodbye.